For centuries, skeptics argued that the Bible was preserved only by tradition. No original manuscripts, no scientific way to compare damaged fragments, no independent method for determining whether two pieces of parchment once belonged to the same scroll. Just a chain of copying, memory, and faith. Then scientists began testing the scrolls themselves. Using the DNA preserved in ancient parchment, researchers have started asking questions that earlier generations could not even formulate. What animal supplied the skin? Did separated fragments come from one manuscript or several? Were some pieces produced in the Judean desert or brought there from somewhere else? And what does the evidence actually tell us about the biblical text? This investigation begins at Qumran, beside the Dead Sea, where 11 caves yielded more than 900 ancient manuscripts and many thousands of fragments. The landscape is severe: limestone cliffs, dry ravines, and air so arid that organic material can survive for 2,000 years. The caves do not look like a library. They look like openings in the rock. But inside them, the history of Scripture was preserved in darkness. The first discovery is the setting itself. Qumran is not an abstract point on a map. It is a real archaeological site on the northwest shore of the Dead Sea, connected to a network of caves in the Judean desert. Pottery jars, textiles, wooden objects, and fragments of parchment were found across this landscape. The scrolls were not discovered in a modern archive. They were recovered from a physical environment with identifiable geography, climate, and material culture. That matters because archaeology begins with context. A fragment removed from its setting can still contain writing, but the site tells us how it was stored, protected, and related to other objects. The caves provide the first layer of evidence: location, preservation, and association. The second discovery is more technical, but equally important. Parchment is not paper. It is prepared animal skin. That means every scroll contains biological information. Even after the writing has faded, the collagen and genetic material within the skin may preserve a record of the animal from which it was made. Researchers do not simply scrape the writing or cut into the most visible letters. They work with tiny samples and contamination controls. Modern DNA is everywhere. It is on our hands, in the air, and in the equipment used to study an object. Ancient DNA research must therefore separate modern contamination from signals that genuinely belong to the parchment. The goal is not to make the scroll speak a sentence it never contained. The goal is narrower and more defensible: to identify biological relationships within a collection of fragments. The third discovery concerns the great Isaiah scroll. The scroll displayed in the shrine of the book is more than 7 meters long and contains the complete text of the book of Isaiah. It is one of the most important manuscripts recovered near Qumran. Its language is ancient Hebrew. Its material is parchment. Its preservation allows scholars to compare a text copied more than 2,000 years ago with the Hebrew form transmitted in later Jewish manuscripts. That comparison does not mean every spelling difference disappears. Ancient scribes used different conventions. Words can be divided differently. Individual readings can vary. But the broad structure of Isaiah is recognizable across the history of transmission. The Great Isaiah Scroll demonstrates that a biblical text existed in a stable, substantial form long before the medieval manuscripts traditionally used by scholars. DNA does not prove that Isaiah was divinely inspired. It does something different. It authenticates the material object as an ancient animal skin manuscript and helps researchers study the physical history of the scroll. Textual comparison then addresses the words. Archaeology addresses the setting. These are different questions, and responsible investigation keeps them separate. The fourth discovery is the unexpected presence of cow skin. In several fragments associated with the book of Jeremiah, genetic analysis indicated that the parchment was made from cattle hide rather than sheep. At first glance, that may seem like a small technical detail. It is not. Cattle require substantial water and food. In the arid Judean desert, they were not the obvious local source for parchment. The result raised the possibility that some scroll material had traveled into the region from elsewhere. This finding does not disprove the scrolls. It changes the question. Instead of assuming that every manuscript was produced beside the caves, researchers must consider trade, movement, and the possibility that communities carried texts across long distances. This is how archaeology works. A surprising result is not a failure. It is a correction to an assumption. The fifth discovery is that DNA can help test whether fragments belong together. Many Dead Sea Scroll fragments are small, damaged, and separated from the manuscripts they once formed. Their writing, handwriting, leather, stitching, and paleography can offer clues. But those clues are not always decisive. Two fragments may look similar because they were produced in the same period. A fragment may be assigned to a scroll because its wording appears to continue a known passage. Material evidence can strengthen or challenge that assignment. If fragments came from the same animal, their genetic relationship may support the idea that they belonged to one skin or one production sequence. If they came from different animals, that does not automatically separate them. A single scroll could be assembled from more than one hide. DNA is therefore one line of evidence among several, not a magical label attached to every fragment. The sixth discovery is the warning hidden inside the science. Ancient DNA is powerful, but it has limits. It cannot by itself tell us who wrote a passage. It cannot reconstruct missing words. It cannot decide whether a theological claim is true. It cannot turn an uncertain fragment into a certain one merely because a laboratory produced a graph. What it can do is expose assumptions. If a fragment thought to belong to one manuscript carries a different biological history, that assignment must be examined. If several fragments share material evidence, scholars have another reason to study their relationship. If modern contamination appears, the result must be treated cautiously. The science becomes valuable precisely when it refuses to say more than the evidence can support. The seventh discovery is the convergence. At Qumran, geography places the manuscripts in a real Judean desert environment. Archaeology identifies caves, jars, settlement remains, and objects associated with the period. Parchment science identifies the animal materials used to create the manuscripts. DNA analysis helps test relationships among fragments and reveals that some scrolls may have traveled from outside the desert. Paleography studies handwriting. Radiocarbon and material analysis help establish age. Textual comparison shows how ancient readings relate to later forms of the Hebrew Bible. None of these lines of evidence, standing alone, proves the entire history of Scripture. That would be an irresponsible claim. But together, they create a far more precise picture than either skepticism or exaggeration can provide. The Dead Sea Scrolls are not modern inventions. They are not a single uniform library produced by one hand. They are a complex collection of ancient manuscripts, copied by different scribes, written on different materials, and preserved in a specific landscape. Some questions remain open. Some fragment assignments are debated. Some popular claims go beyond the evidence. But the central fact remains. Biblical texts were circulating in substantial written forms more than 2,000 years ago. The physical manuscripts can be studied. Their materials can be tested. Their words can be compared. Their archaeological context can be investigated. That is why the DNA findings matter. They do not replace faith with a laboratory result. They do not remove the need for interpretation. They do something more valuable. They force every interpretation to answer to the material record. Let us be rigorous. Let us examine the alternative explanations. Some fragments were moved. Some were copied more than once. Some may have been misidentified. And yet, across geography, archaeology, parchment, genetics, handwriting, and textual comparison, the evidence continues to converge. This is not speculation. This is not interpretation. This is what the earth has surrendered. If this investigation has challenged your assumptions or strengthened them, enlist in our next expedition. Hit the subscribe button right now. Our next investigation goes where the evidence leads. The ground keeps confirming the book. I am your guide through the evidence that connects scripture to soil, belief to bone, and history to the artifacts buried beneath our feet.